It's my pleasure to introduce the next speaker. He really doesn't need any introduction at all, being past president of the association. So Professor Sir Munir Pirmohamed is from the University of Liverpool, and he's going to be talking to us about one of his passion, pharmacogenomics, from drug discovery to implementation.
Thank you very much. Thank you for the introduction, and Claire, thank you for the invitation to give a talk, and thank you for organizing this fantastic meeting. The thing I like about the AOP is that it's not only about breadth, but also depth as well at the same time, and it's a great learning experience. So I'm going to give you a talk on this area. It's a broad area, and I'm going to give you a snapshot in terms of what's going on in this area. So in 2023, I wrote a review article for Nature Reviews Genetics, and this is the definition that I used for pharmacogenomics in that review article. It covers two areas where you can use genomic technologies to enable discovery and development of novel drugs, but also how you can look at optimizing the drug dose and choice to overcome some of the variability issues that we see in everyday clinical practice. And I'll cover both of those. So have you ever wondered how much of the genome is actually druggable? So we worked with Chris Finnan and Aaron Hingorani at UCL to be able to look at this. And of the about 20,000 genes in the human genome, about 24% are said to be druggable. That's probably an underestimate as based on current technology. And as new technology develops, advanced therapies and so on, it is likely that the number that will be druggable will increase. But if you just look at that 24% that are druggable, about 74% remain undrugged at the moment. So there's huge opportunities in the genome to develop new drugs, identify new drug targets. And you can see this from work that people have done. This is a review from Nature from Vincent Muser, who was able to show that 40 germline genetic observations have led to new targets and approval of therapies. The median time between target and genetic discovery has been variable, up to 25 years, but with the technologies we have now, the time span is getting shorter. And most of the therapies have been there for rare diseases that compensate for loss of function mutations. Last year, Matt Nelson looked at the impact of genetic evidence on clinical success in terms of drug development. As you know, drug development is a risky and expensive business, and with many failures, perhaps 90% of the drugs that go in terms of development fail to make it to market. And clearly, if you look at the overall expense, there are estimates from 1 billion to over several billion in terms of cost of bringing a drug to market. So people have been looking at genetic data to be able to see how you can improve success for developing drug. Matt was able to show the probability of success for drug mechanisms with genetic support was 2.6 times greater than those without, which is really important. And he shows the success improved with the confidence in the causal gene, but it wasn't affected by genetic effect size. Many of you are working on GWAS, where you have relative risks of 1.2, 1.4. That doesn't mean that it may not be, that still means that something you may have discovered may still be an important target that you can actually look at in terms of new drug development. It was also unaffected by minor allele frequency and the year of discovery and so on. So this is a really important area, and I think most drug companies are now using genetic data to be able to identify potential new drug targets. Perhaps the best examples of that, and the best known example, is with PCSK9 inhibitors. Based on genetic data, people have shown loss of function mutations, lower plasma levels of LDL-C, and lower cardiovascular disease risk. Monoclonal antibodies have been developed and are now in clinical practice with long-term clinical data highlighting effects on cardiovascular outcomes. More recently, people have also been looking at angiopoietin-like 3, which is used for mixed hyperlipidemia, or at least potentially for mixed hyperlipidemia. Inhibits lipoprotein lipase and endothelial lipases, and loss of function variants decrease LDL-C levels and decrease cardiovascular risk. There is a monoclonal antibody at phase 1, and this week in The Lancet, an siRNA phase 2 trial which showed reduction in ApoB and reduction in hepatic fat. So really interesting things which are happening in the cardiovascular field in this area. However, even when you look at these particular trials, you still see quite a lot of variability, even with the PCSK9 inhibitors and with the angiopoietin-like 3 inhibitors as well. And people are starting to see how you can actually try to use genomics data to be able to identify those who are more likely to benefit from these drugs, because these drugs are expensive and we may need to stratify in clinical practice as we move forward. So here's something on PCSK9 inhibitors. Comes from the Odyssey trial, which had about 12,000 patients involved. They were able to derive a genome-wide polygenic risk score comprising about 6.5 million SNPs. And from that, they were able to identify those who are high risk and low risk of coronary artery disease, and able to show, as you can see on the Kaplan-Meier plots, that those at high genetic risk were more likely to benefit from this PCSK9 monoclonal antibody. The absolute relative risk reduction was 6% in the high-risk group compared to 1.5% in the low-risk group. So really interesting things which are going on, and we're working on particular targets based on genome-wide data and so on, and many other people are doing that. It's interesting to note that where you're getting success, more likely to get success where you have diversity of data. If you're just working on one particular ancestry, perhaps reduces the chances of success. But if you have much more diverse data, then you're much more likely to get success in trying to in identifying a particular target that you can pursue. Sorry. Okay, let me move on to the next aspect, which is actually how can you use pharmacogenomics to be able to improve efficacy and safety of drugs that we already have on the market. If you look at variability in drug response, there's huge variability in drug response in many of the drugs that we use, in fact, in most of the drugs we use. So if you look at efficacy, 90% of drugs only work in 30 to 50% of patients. And what happens is you give a drug, you wait for the response, that may take 3 months, it doesn't work, you start another drug, etc., and so on. So a trial and error approach is not good for the patient, it's not good for the health economy. In terms of safety, 6.5% of all admissions to our hospitals are due to adverse drug reactions. At this very moment in time, there are about 8,000 beds in the NHS which are occupied by patients with adverse drug reactions. At a huge burden, a cost to the NHS. So we've estimated that the burden of adverse drug reactions causing hospital admission is about £2.2 billion per year. And we need to do something about that, important for the economy, but also for the actually patient outcomes as well. And there are many examples now out there. And as I have worked in this particular area, I've learned that my work was mostly translational initially, but then we've identified particular genes that you can take into clinical practice, and we've had to move some of our work to more application and then actually implementation as well. And you need different skills and you need different collaborators to be able to take that into clinical practice. So I'm going to give you some examples and then tell you where we're at right now. So if you look at drug hypersensitivity reactions, these are reactions where you might get Stevens-Johnson syndrome, toxic epidermocrolysis, drug-induced liver injury. Two examples that we worked on. One is in carbamazepine, which can cause these serious hypersensitivity reactions. And with only 22 patients with hypersensitivity syndrome, we were able to get a genome-wide significant result. We were able to localize this to an HLA allele, A*3101. This has now been replicated all over the world, and it is in the drug label. There's another allele in a different ethnic group in Southeast Asian, which is HLA-B1502, which is also in the label. And those two alleles are potential for implementation into clinical practice. So we've put in an application to the genomic test directory that everybody in this country who requires carbamazepine should have those two alleles tested for before they get carbamazepine. Carbamazepine is very rarely used in epilepsy now, but actually the usage is mostly by dentists for trigeminal neuralgia. And so we need to be able to upskill dentists in terms of using genomic testing as well. Another example is allopurinol, still very widely used for gout treatment. It causes serious cutaneous adverse reactions, and SCAR stands for that. This is unpublished data in only 13 cases of SCAR, again showing a genome-wide significant result with 13 cases localized to HLA-B star 5801 with an odds ratio of 35.9, while in diabetes you're looking at a relative risk of 1.2, 1.4. We are managing to find odds ratios of 36, etc., based on very small numbers, almost like a Mendelian disease, which highlights the possible potential for implementing this before we actually start giving patients these drugs. So what's around the corner coming into the NHS this year and over the next few years? Well, the first example is clopidogrel. Many of you will have prescribed clopidogrel. It's a drug which is used in coronary artery disease, cerebrovascular disease, peripheral arterial disease. It's a prodrug. It needs metabolism in the liver by a P450 enzyme called cytochrome P452C19 for activation to its thiol active metabolite, which then inhibits platelet aggregation. Theoretically, one would expect that if you can't activate it, then you'd get less benefit from that, and that's been proven to be true in many different studies. So here's a study from Jessica Megger, published in the New England Journal of Medicine, where they looked at stent thrombosis following with the use of clopidogrel. And what they found was those individuals who were carriers of a loss-of-function allele were much more likely to get stent thrombosis. Now, in cardiology, it's not been taken up. There are many reasons for that, which I won't go through at the moment. But in stroke medicine, it is starting to be used. And NICE has done an assessment of this. It was published in July last year. They looked at the clinical effectiveness and the cost effectiveness of genotyping people for cytochrome P452C19 before you put them on clopidogrel for either for TIA or ischemic stroke, and they showed it to be clinically effective and cost effective. And this is about to be implemented in the whole of the NHS. It's already being done. There's already studies, or there's already service in Tayside in Scotland. There are 4 pilot sites at the moment in England which are testing this, and very soon, hopefully, it will be available for anybody who requires it for stroke or TIA. It's estimated the 100,000 people in England itself will require CYP2C19 genotyping before they go on clopidogrel when it's introduced. The important thing, and it's becoming increasingly clear, is to think about the diversity aspects and the variation in polymorphism frequency according to ethnicity. So if you look at cytochrome P452C19, it is much more prevalent in South Asian populations. So this is data from East London Genes and Health, and you can see that the loss of 2 cytochrome P452C19 alleles was much greater in that population compared to the European population, and there were much higher risk of developing recurrent myocardial infarction when treated with clopidogrel. So by not genotyping, we may be exacerbating health inequalities and so on. So let me just move on to the next thing which is coming through, which is fluoropyrimidines. 5-fluorouracil, widely used in about 15% of cancers, causes severe toxicity in 10 to 30% of individuals, and mortality in about 0.5 to 1%. In October 2020, following a study which was undertaken in Netherlands, which showed that if you can genotype for these particular polymorphisms in DPYD, you can reduce toxicity associated with 5-FU. And in October 2020, the NHS introduced these into practice. And within a year, went from 0 tests to about 38,000 tests a year in England only, about 45,000 tests overall in the UK. Problem with this is that it's all derived from European ancestry population. About 17% of the population of the UK is non-European ancestry. So what happens in them? So we've been looking at that and we identified many different variants in non-European populations. One particular variant, which we feel very important, is this particular variant, which is found in African ancestry individuals. It is associated with a reduction in enzyme function. It's found in 2% of African reference populations. And it is in fact included in DPYD testing panels in several laboratories in the United States. And in the UK, 4.2% of our population is from that particular ancestry group. And so we made an application to the Genomic Test Directory for inclusion of this particular variant into our DPYD panel, and decision is awaited. But we also worked with the chemotherapy board, and this has now been included in the guidelines for use of 5FU. We are still working on identifying additional variants in DPYD, undertaking UK-wide recruitment, identify people who've already been genotyped but still get toxicity, grade 3 plus toxicity. And we so far, in 187 patients, identified 49 novel coding variants. The problem is, what is the function of that? And so we are developing functional assays to be able to look at that, as well as some PBPK modeling as what to do with the dose. Most of the patients we're recruiting in the UK are Caucasians, 97%. So we are also doing international recruitment to be able to look at the diversity of changes which occur in the gene, and then see, make sure that we actually cover the whole of our diverse population. Again, during the pandemic, working with Donald O'Donoghue in this college, we set up a working group to look at pharmacogenomics, and we published a report called Personalized Prescribing. And this was published about 2 years ago. And in that, basically we said that it is now prime time to actually introduce pharmacogenomic testing into the NHS. And we advocated panel testing. There are many issues in terms of implementation of this, including the lack of skills in our workforce in this area, so we need a comprehensive educational package. But it's also important to have that clear lines of communication with the public and patients in terms of the benefits and the limitations of pharmacogenomic testing and so on. We advocated the use of panel testing for pharmacogenomics. At that time, there was very little data on that. Last year, we published a trial in the Lancet of a 12-gene pharmacogenetic panel which had 50 different variants on that panel. This was a cluster randomized crossover implementation study undertaken in 7 European countries. Liverpool was the only UK site, and we recruited about 6,900 patients, half in each group. And we showed that that 12-gene panel, which cost less than 50 quid, was able to reduce adverse drug reactions by 30%, which is a huge reduction based on such a simple test and very cheap test as well. So what's going on in terms of panel testing in this country? Well, there's a pilot study which is going on, led out of Manchester in the northwest and in Liverpool, which is called the Progress Study. It is based in primary care, and it's focusing on 5 medicine classes, as you can see there. It's not powered to address utility, clinical utility, but it addresses the performance of the testing pathway and the adherence to guidance and prescribing modifications. And the pilot, the data so far is in 40% of cases, recommendations will switch to the drug or change the dose, and the compliance to the guidance by the general practitioner was about 90%. In order to be able to help that implementation as well, we've just been awarded a UK Centre for Excellence in Regulatory Science and Innovation in Pharmacogenomics. Many different work packages there, including developing guidelines. The first guideline we are doing is on clopidogrel and 2C19. Work package 2 focuses on industry testing of pharmacogenetics. There's an awful lot of companies which are offering the public pharmacogenomic tests at the moment. All the DNA from those tests actually goes abroad to the United States, to China or India, which seems a bit strange to me, that a country that invented sequencing, all the DNA is actually going abroad. I'm sorry, I sound like Trump, protectionism and so on. I apologize, I'm not meant to be like that. But I think we should be able to have a better regulatory pathway here in this country that allows industry to grow to do the pharmacogenetic testing here. And so the aim of the CERSI is really to develop proportionate, forward-looking, inclusive and responsive regulatory pathways that enhance innovation. We're working with MHRA, with NICE and various other bodies to be able to do that. So let me conclude. I've just run out of time, sorry, I think. So pharmacogenomics has been applied to both drug discovery, development, and optimization responses. Target identification is really important. I think that we need to move from doing single gene variants in the pharmacogenes to actually looking at the variation across the whole gene, and DPYD represents an example of that. Implementation programs are underway, but the challenge is scalability. How do you scale to 68 million people in the UK? We may need 1 million more tests per year if we actually introduce the panel testing in the UK, which requires a huge amount of effort and capacity to do that. It's important as we move forward that we look at different ethnic groups and we do not exacerbate health inequalities. So I just want to thank my co-workers and thank you for your attention.
Thank you. Your passion has really shined through. Shall we take a couple of very quick questions, even though we're slightly over time? Are there any quick questions? If not, I have a burning one. Given what you said about pharmacogenomics and the development of different polygenic risk of many different diseases, do you see in the future we can sequence everybody in the NHS and then send it to a central place where we can ask questions of what drug to prescribe, what at the right dose, and perhaps at what risk of what diseases?
Yeah, so ideally, I think that will happen eventually. And many people will have whole genome sequences done, and the data, pharmacogenomics data, will be extracted from that. Obviously, there's already a newborn sequencing program going on in this country, and eventually every child born in this country will have a whole human genome sequence done at birth, and we can extract that data and make that available. The critical issue is actually integrating that data into the electronic health record and making sure that we can access that data. When I did the PREPARE trial in Liverpool, I said, I want to integrate that into our electronic health record. And they said to me, they can only have a PDF, which is not machine readable. And that's a problem.
One question from Daniel.
Excellent talk. Do you think an argument can be made that all phase 3 pivotal studies that are randomized should have whole genome sequencing of all participants so that we can, before we pick up adverse events, we can detect the strongly genetically determined events?
So I think all phase 3 trials, academic and industry, should be collecting DNA. Not necessarily do whole genome sequencing, but they should have that DNA that you can do whole genome sequencing or panel testing and so on. Because I think particularly with polygenic risk scores, we need to be able to test them in trials. And we're not going to be able to do that in some of the observational bioresources that we have. Safety maybe is different. You can actually, because of the larger effect size, but for efficacy, I think we're going to need trials to be able to do that. And we won't be able to afford specific trials to be able to do that for polygenic risk scores. But therefore, if we collect DNA, which is available to do that, then it allows us to be able to look at polygenic risk scores in that situation.
Last question at the back.
Munir, lovely review. Stroke, clopidogrel. 25% of the Western population are loss of function in this country. So every time we give clopidogrel, which as you said is our first line therapy after an ischemic stroke or TIA, we have a 25% chance of giving chalk and not an active drug. We desperately need to test. We have point of care devices to do this. We have been told very clearly there is no money for this, and when you look at them and raise your eyebrows, they say, well, what do you want us to stop doing? So we are going to go into a position where we know what to do and we can't afford to do it, and people will have stroke recurrences.
Yeah, no, and that is a challenge. There's a challenge of actually undertaking the genomic testing at scale, but also some of the costs associated with that. But it is cost effective, and it is coming actually. And I think that NHS in England at least is looking at that, and Scotland, they're about to implement that as well. It's already implemented, as I said, in Tayside. And with pharmacogenomics, the major issue is going to be the cost outlay, even if we can show cost effectiveness. And that is going to be a challenge. And we need to be able to persuade our politicians that it is worth doing this, to be able to implement this, to improve the outcomes. If Wes Streeting talks about prevention, actually doing genomic testing is prevention. As any prescriber in this room, you look at all information you have on the age, what other drugs they're on, liver function, renal function, and pharmacogenomics is just additional data, additional information to be able to improve what you do in terms of prescribing. And it is all part of prevention.
Thank you.
Great.